Hello. Hello. Can anyone hear me? Okay, so let's wait for 5 more minutes till other people are joining. Hello. Uh, I Let me introduce myself. I am Nilanjana. I am doing my PhD in immunology and virology. So I am a TA under Professor Himanshu Kumar. So you guys also can introduce yourselves, from where you are and why you took this course. So Simran, you can tell. You guys can also unmute yourself and tell.
So, hi, this is Eldon from Hosur, and I'm doing my MSc in biotechnology, ma'am.
Okay. From where?
Hosur,
Okay. MSc in biotechnology. Okay. Which college?
MGR College.
Okay. And Simran?
Hello, ma'am. I'm Simran Khanam from Bardhaman in West Bengal, and I am Masters MSc in Nutrition and Public Health uh, from Rani Ashwini University.
Okay. So why you took this course, Simran? Hello? Um, so, hello, can you hear me?
Yes, ma'am, we can hear you.
Why you took this course?
Basically, I have studied immunology in my BSc and MSc also, ma'am. I just want to know in depth about the host and pathogen relationship. That's what.
Okay. And Simran, okay, she left. So are you having any doubt, like currently in the last 2 weeks course?
No, ma'am. As of now, I'm okay with the concepts.
Okay. Okay, I, uh, uh, so I will teach today, like what sir has taught in week two. Some more like same thing only I will teach in a more elaborate way. So uh, today I'll be teaching about the cells and organs and microenvironments of the immune system. Can you see the slide?
Yes, ma'am.
Um, okay. So first, before going on to what are the cells and organs, so have you seen that how the cells are generated? Like, what is the process by which these cells are generated? Hello, Simran.
ma'am. Sorry,
Uh,
ma'am. Actually, some network issue. That's why I leave this page.
okay. So can you see my slide?
Yes, ma'am.
So you
Um,
are having any
no, no,
doubt?
no, no, no. Sorry, ma'am. Sorry, ma'am. I can see your page. I can see your slide.
You can no.
No,
You cannot.
no, ma'am.
Okay, now you can.
Yes, ma'am.
Okay. So Simran, you are having any doubt? Like in week 1 or week 2, what sir has taught? Any kind of doubt?
Hello.
Yes, yes. Hello. Yes, Simran, you can tell.
No, ma'am. I have no doubt.
Okay. Okay, so today I will just go through what sir has taught already. So first, before going on to the cells and organs of the immune system, I will first tell how the cells are coming up. What is the process through which the cells are coming up? So, do anyone know what are stem cells? Any of you? So, in our immune system, the cells are generated through a process known as hematopoiesis. So, before going on to what is hematopoiesis, first we should know what are stem cells. So, stem cells are cells which are having two properties. Like one, they can self-renew or regenerate, and second, they can differentiate into different types of cell types. So, in our body, all the mature blood cells, including the red blood cells, granulocyte, macrophage, dendritic cells, lymphocytes, arise from a single cell type, that is a hematopoietic stem cell. Hematopoietic stem cell is the mother cell from which these cells are arise. So, the process by which this hematopoietic stem cells differentiate into mature blood cells is called hematopoiesis. So, the process is known as hematopoiesis. So, there are different types of cell types in our body which can give rise to other cell types. Like, for example, there are pluripotent stem cells, there are multipotent stem cells, and there are unipotent stem cells. So, pluripotent means they can give rise to an entire organism, like a fertilized egg, the zygote. It can give rise to the whole organism. So, it is an example of a pluripotent stem cell. And there are multipotent stem cells, which arise from the embryonic stem cell and can give rise to a more limited range of cell type. Like not the whole organism it can give rise to, but some of the cell type it can give rise to. And other is the unipotent stem cell from which we can get only a similar type of cells. Here we can see that there are different meanings of pluripotency, multipotency and totipotency. So totipotent means in the initial stage, this cell can form the all the cells of the body as well as this embryonic membrane. Like during the fertilization process, this development of modula, blastocyst is an example. Like these are totipotency or totipotent stem cells, which can give rise to the whole, like the along with the cells, it can give rise to the membranes also. And there are pluripotent cells which cannot form the extraembryonic membrane, but it can give rise to an entire organism, like I told, that zygote. And others are these multipotent and unipotent cells. Like these pluripotent cells can give rise to specific type of cells which can again give rise to a more limited range of cell types. Like some cells can form this digestive tissue, some can form this nervous tissue, some can form this cardiac tissue. And unipotent means only a specific type of cell they will give rise to. So, in our body, the hematopoietic stem cells differentiate into all red and white blood cells. So, first, these hematopoietic stem cells originate in the fetal tissue and they are primarily residing in the bone marrow of the adult vertebrates. And a small number can be found in the adult spleen and liver. So, it is very rare. These hematopoietic stem cells are very rare. Like less than one hematopoietic stem cell is present per 5 into 10 to the power 5 cells in the bone marrow. So when our immune system is challenged by any pathogen, then only this HSCs divide. Otherwise, when it is like in a normal state or a steady state, these are mostly in a quiescent stage. That is, they will remain normal. They won't generate any more cells and only a small number will divide, generating daughter cells. So our body needs to be invaded by pathogen to make it active. So some daughter cells retain the stem cell characteristics of this mother cell, that is, they remain self-renewing and are able to give rise to all blood cell types. Other daughter cells can differentiate into progenitor cells that have limited renewal capacity and become progressively more committed to a particular blood cell line, like multipotent cells. And if it can give to all blood cell types, then it will be pluripotent cells. So as an organism ages, this number of HSCs decreases, demonstrating that there are limits to an HSC self-renewal potential. So as an organism ages, also our there is a decrease of this HSC, so our immune system also becomes weak. So when there is an increased number of demand of hematopoiesis, for example during an infection or after chemotherapy, these HSCs will proliferate rapidly. So the transplantation of HSC is an important therapy for patients whose hematopoietic systems must be replaced. Like in some patients, like for example, if some person is suffering from a disease known as severe combined immunodeficiency, where this person's immune system is very weak and cannot perform all the activities, then that person needs to have to provide a functional immune system to that person. These HSCs can be transplanted from a normal person to that person so that they will have a normal immune system. Or otherwise, if a person is suffering from any disorder like sickle cell anemia or thalassemia and not producing enough blood cells, then we can replace this defective hematopoietic system with a functional one to cure patients with these disorders. Also, people who are suffering from cancer, they also can be substituted with a new kind of hematopoietic stem cell. So in immune deficient patient, we can just we can substitute that hematopoietic system with a new hematopoietic system so that the immune system will become active in that patient. So there were early approaches were there to isolate hematopoietic stem cell. They took the advantages of antibodies that were raised against mature blood cells in a process called panning. So what they did was they have a plastic in which they coated a suspension of bone marrow cells and they put antibody that would bind to different types of proteins which are present in these cells. Like all these HSCs or the hematopoietic stem cell will have markers on them. So earlier, when they wanted to isolate these HSCs, what they will do, they will use antibodies which are specific for these markers, and they will coat this plastic with the antibodies, and they will first lay a suspension of this bone marrow cells, and they will put the antibodies over the suspension. What will happen? The antibodies will bind to that specific cells which are having those specific markers. Here we can see that lean plus or lineage positive cells are the hematopoietic cells. So the antibodies will bind in this. So there are different types of cells. Here we can know this S is the stem cells, and uh, this stem cell actually lack these markers. So these antibodies, other antibodies like the eosinophil, neutrophil, other blood cells, they will have this marker, lim marker, and these antibodies will bind to these cells. So these cells, we can see that the antibodies are binding to the cells. So we can discard what does not bind and keep what does bind. So here we can see that these cells are binding to these antibodies. So these are having that lean plus, that is the lineage positive cells these are. And what doesn't bind, we can isolate, and from there they could isolate these hematopoietic stem cells from here. That is earlier uh, they did this experiment to isolate the hematopoietic stem cell. This is a microscope image. So what uh, was as I told this experiment. So similarly, what they did was they did a negative selection against cells that express the lineage markers, that is the markers which are present in the cells. And here they did a positive selection, means that in which there were no uh, like markers of the lineage marker, but there were presence of other markers which are present on the stem cell. So they separated these cells based on the markers, and what they did was they took mice and they lethally irradiated the mice with radiation, so that like the mice become immunodeficient. So after that they put these cells into the mice to see which cell is actually restoring this hematopoiesis. Like this, they came to know that yes, these cells are actually important for this hematopoiesis process, and others are not involved with this hematopoiesis process. So by using antibodies in the earlier days, they found out that these hematopoietic stem cells can actually play a role in development of these immune cells. So anyone, if you guys are having any question, you can raise your hand and ask me in between. So this hematopoietic stem cell can differentiate into both myeloid cells as well as lymphoid cells. So what are myeloid cells and lymphoid cells? So there are two types of hematopoietic stem cell. One is the long-term hematopoietic stem cell, and other are the short-term hematopoietic stem cell. And long-term means which are long-lived and they do not divide rapidly. And short-term means which have a more proliferative multipotent progenitor. They will divide rapidly and form this lymphoid and myeloid cell types. So myeloid progenitor cell means they will give rise to the red blood cells, platelets, and myeloid cells like granulocyte, monocyte, macrophage, and some dendritic cells. While lymphoid progenitor cell means they are known as common lymphoid progenitor or CLP, and they give rise to B lymphocyte, T lymphocyte, innate lymphocytes, as well as specific dendritic cell population. So this hematopoiesis process is regulated by a number of transcription factors. So different types of transcription factors helps in the development of this myeloid progenitor and lymphoid progenitor. So here we can see this long-term HSC hematopoietic stem cell give rise to the short-term hematopoietic stem cell, which have the capability to proliferate rapidly. And from this myeloid progenitor, this short-term hematopoietic stem cell then give rise to the uh, another type of cells which can give again rise to this myeloid progenitor and this lymphoid progenitor. So these transcription factors will play an important role in determining which lineage it will go, whether it will go to myeloid progenitor or it will go to lymphoid progenitor, and they will then again divide it to granulocyte. This myeloid progenitor will give rise to the granulocyte monocyte progenitor and this erythroid progenitor, while this lymphoid progenitor will give rise to T cell progenitor or B cell progenitor or dendritic cell. So all this hematopoiesis process is regulated by the transcription factors. So cells of the myeloid lineage are the first responders to infection. So this myeloid lineage, which include the red blood cell, granulocytes, monocyte, and macrophages, will first respond to infection whenever there is an invasion of pathogen in our body. So here we can see this myeloid progenitor is giving rise to monocyte, basophil, eosinophil, neutrophil, and this monocyte again gives rise to macrophage and dendritic cell. So white blood cells within this lineage are innate immune cells that respond rapidly to the invasion of a pathogen and communicate the presence of an insult to cells of the lymphoid lineage. So whenever there is an infection, these white blood cells will come into the picture and they will rapidly respond to the infection and they will help in communicating this signal to the lymphoid cells of the lymphoid lineage. So basically granulocytes are the first responders during an immune response. And why they are known as granulocytes? Because they are having they differ in their properties by their presence of the cytoplasmic granules, and they also release these granules whenever there is a invasion by the pathogen. They will release these granules so that these granules will attack the pathogen and clear the infection. And these granulocytes have multi-lobed nuclei that will make them visually very much distinguishable from the lymphocytes whose nuclei are round. So here we can see they are having multi-lobed nuclei, and there are presence of the granules inside these cells which will be released during when there is a pathogen. And here there are different types of granulocytes like neutrophils, basophil, eosinophil, mast cell. So these neutrophils are the majority and they constitute about 50 to 70% in adult humans. And compared to these neutrophils, there are less number of eosinophils, basophils, and mast cells. Basophils and mast cells are almost less than 1%, and eosinophil is 1 to 3%. So here we can see that these all are having almost a similar property, like they are having bilobed nucleus and presence of granules, and due to their staining property they will be differentiated into eosinophil, neutrophil, or basophil. So there are different functions of these granulocytes. Like for example, the neutrophils will kill the bacteria, fungi, and foreign debris. These are the examples of the molecules which are present in the granules, like proteases, antimicrobial proteins, protease inhibitors, histamine, which are present in these granules. And the examples are the protease contains the elastase or collagenase, or antimicrobial proteins include defensins, lysozyme, or antitrypsin, and their function is like tissue remodeling, direct harm to pathogen, regulation of proteosis, vasodilation, and inflammation. While in case of eosinophil, we can see they are also having this cationic protein, ribonucleases, cytokines, chemokines, and these are the example of this like cation proteins like EPO, MBP. These are the different types of proteins which are present in these granules. And the cytokines include IL-4, IL-10, IL-13, TNF alpha. Then the chemokines include RANTase, MIP1A, and these all are mainly involved in induction of the formation of reactive oxygen species, vasodilation. They are having some antiviral activity, modulation of adaptive immune response, and they also can attract leukocytes. So mostly eosinophil are increased, as I already told that the level of eosinophil is 1 to 3%. But while there is an allergic response in our bodies, this eosinophil will increase in number, and they will perform the activities to this. They will secrete these proteins from the granules, and that will help in reducing the allergy. So the other things are basophil and mast cell, which also uh, contains the granules contain cytokines, lipid mediators, and histamine, and they can modulate adaptive immune response, regulation of inflammation, vasodilation, and smooth muscle activation. So basically, basophil also is involved in allergic response, like eosinophil. So their number will increase when there is a allergy allergic response which is there in the body. So apart from that, as I told, like monocytes and lymphocytes, they are also these monocytes will clean up the damaged cells. Lymphocytes will help fight viruses and make antibodies. But this is not in the granulocytes; they are different. So there is a process known as leukocytosis where there is a abnormally large number of leukocytes usually associated with acute infection. So when there is an infection, there can be an increase in the number of this leukocyte, and if there is a too much increase in the number of leukocyte, then it can be from the blood test. You can identify that there is an infection inside your body, and so the body has increased the number of leukocytes, and the count greater than 10,000 millimeter cube may be considered as leukocytosis. So here we can see like basophil, eosinophil, neutrophil, everything is increased here. High WBC count we can see. But here it is low WBC count. So normally the WBC count should be low only. Only in presence of infection, it can increase to uh, like count greater than this 10,000 millimeter cube, and this condition is known as leukocytosis. So at the site of infection, these molecules will come, and then they will release these granules which help in clearance of the pathogens. So any doubt till now? Anyone can like raise the hand and tell if any doubt is there. And other people has also joined, so I am asking again whether last the two in the like the talk in you have any doubt for last week one and week two. If there is any doubt for week one and week two, you can ask me. uh, so I guess no doubt is there for week one and week two. So okay, so let me then continue. If anyone is having any doubt, you can raise your hand and ask your doubt. So after that, we come to the antigen presenting cells. So, uh, like what are antigen presenting cells? Uh, they are the cellular bridge between innate and adaptive immune system. So they kind of connect this innate as well as adaptive immune system. So there are three major activities they do when they encounter the pathogens and they become activated. They secrete some proteins that attract and activate other immune cells. They internalize pathogens via phagocytosis. They digest pathogenic proteins into peptides and then present these peptides antigens on their membrane surfaces via MHC class 2 molecules. And they upregulate other co-stimulated molecules required for the optimal activation of helper T cells. So, the antigen presenting cells include this monocyte, this macrophage, dendritic cell. All these cells will consist of these antigen presenting cells, as well as B cell also is considered as a antigen presenting cell. So they basically will secrete some proteins like the cytokines, chemokines, they can secrete and they can activate other immune cells. They can also internalize pathogens via phagocytosis. These macrophage cells will do this phagocytosis where they will process these pathogenic proteins into peptides and they will present these peptides. Why it is called antigen presenting cells? Because they will present these antigens in and into the like they will present these antigens via there are some membrane receptors present which are known as MHC molecules, major histocompatibility complex. So these molecules are present. What these antigen presenting cells will do, they will digest the pathogenic proteins into small peptides and will present the antigens via these MHC molecules which are present on on the surfaces of these molecules to the T cells or the T helper cells or the cytotoxic T cells. And via that, they will communicate between the, as it is said, like they will make a bridge between the innate as well as the adaptive immune system. And they will also upregulate the co-stimulatory molecules required for the optimal activation of helper T cells. So there are some molecules which are present on the surface of these T cells, T lymphocyte cells. So they will upregulate some co-stimulatory molecules which are required for the activation of these T cells, and then further the downstream post process they will also help in the upregulation of these co-stimulatory molecules. So here we can see these are the monocytes. Here these are the like the microscopic images. These are the macrophages and the megakaryocyte which will give rise to platelets. And this is the dendritic cell. So here you can see this, like there are lysozymes and there are pseudopodia, which I told already that macrophage will internalize the pathogens via phagocytosis, where this pseudopodia will go around the pathogens and it will digest that pathogen and will take inside the pathogen and digest these pathogenic proteins into peptides. And this lysosome will also release some enzymes, the lysosomal enzymes. There are phagosomes also, which will help in this phagocytosis. And when this phagosome and lysosome will fuse, they will form a phagolysosome. So here we can see that all these cells are having this pseudopodia and phagosomes, lysosome, and they will release some enzymes or some other components like cytokines or chemokines, and that will either activate the other immune cells or will clear the proteins, the pathogenic proteins from the cells, or they will engulf the pathogen inside and will digest the proteins into peptides. And via the presence of this receptor, that is the MHC molecules, they are present on this membrane surface, they will help in communication between the to the T. They will communicate to the T cell that there is an infection inside the cell. So thus they will present the antigens and activate the adaptive immune response. So here we can see that there one such cell types is the monocyte, which I already have shown. These monocytes uh, will be activated, and the stimuli are the cytokines, chemokines, or the ligands of the TLR or the toll-like receptors. Uh, this monocyte will get differentiated into macrophage or dendritic cell. So this macrophage have the the function of cytotoxicity or fibrosis, as well as phagocytosis, chemokine cytokine production, tissue surveillance, and this dendritic cell have the function of antigen presentation or T cell activation. So these monocytes, there are different kinds of monocytes, and these monocytes, uh, when they will enter the tissue, they will get differentiated into macrophages, and this tissue will, these macrophages will then uh, go inside the whole, like surveil the, it will, there will be surveillance by these macrophages for the pathogens, and they will then digest or secrete the phagocytosis or secrete the cytokine or chemokine, and they will try to kill the pathogen. And similarly, the dendritic cell mainly helps in antigen presentation as well as T cell activation. So, depending on where these macrophages are, these macrophages can be divided into different types. Like, for example, in the brain, the name of these macrophages are microglia. While in lung, you can, these macrophages are called alveolar macrophages. Then in liver, it is known as Kupffer cell. Then in kidney, it is like resident kidney macrophage. So according to the tissue type, these macrophages will have different types of names. Like, for example, in brain, it helps in the neural circuit development, and the function also changes according to the tissue type. In lung, they will remove the pollutants, microbes, and clear surfactants. In liver, they will scavenge the red blood cells and clear particles. In kidney, they will regulate the inflammatory responses to antigen filter from blood. In skin, it is known as Langerhans cell, and it has skin immunity and tolerance. In spleen, it is known as a red pulp macrophage. It will scavenge the red blood cells, recycle iron. In peritoneal cavity, it will maintain the immunoglobulin A production. In intestine, there will be it is lamina propria macrophage and intestinal muscularis macrophage, and it function is for gut immunity and tolerance, and regulating the peristalsis movement. In bone marrow, it is bone marrow macrophage. It helps in the blood cell development, clear neutrophils, lymph node. It is known as subcapsular sinus macrophage. It will trap antigen particles. In heart, it is cardiac macrophage. It will clear dying heart cells. So you can see so many types of macrophages are there, and it is having so much diverse function in different types of tissue. So these macrophages play an important role in uh, various processes in our immune system. So coming down, there are other cells, as I told, there are erythroid progenitors which are developing from the myeloid progenitor cells, which will which will give rise to the erythrocyte or red blood cells. These erythroid cells contain high concentration of hemoglobin and circulate through the blood vessel and capillaries, delivering oxygen to surrounding cells and tissues. So these erythroid cells also express the surface receptors for antibody, and they bind these antibody complexes that can be cleared by the many macrophages that scavenge erythrocytes. So mostly the function of these erythroid cells are like they help in delivering oxygen to and from the cells and the tissues, and they sometimes play some immune role also, like binding. They are having some surface receptors which will bind with the antibody complexes like the antigen antibody complexes, and these can be cleared up by the different types of like macrophages, like the mm, macrophages which will scavenge these erythrocytes. So they also generate sometimes some compounds like nitric oxide, and that will directly affect the microbes. So apart from that, there are megakaryocytes which will give rise to the platelets. So they are why they are called karyocytes, because the size is very much large, and they will reside in the bone marrow, and they will give rise to thousands of platelets. And we all know that platelets are uh, cells which will help in the formation of blood clots. So they are very small cell fragments or small cells that will circulate in the blood and help in the formation of blood clots uh, during an injury. So here we came to know about the myeloid lineage. So any confusion regarding the myeloid lineage? Okay, then we can now uh, learn about the lymphoid lineage. So, as I already told that the cells of this lymphoid lineage will regulate the adaptive immune response, while that of the myeloid lineage will mostly regulate the innate immune response. Like they will quickly act on the pathogen, but this lymphoid lineage will take time, and uh, like it will take time and will help in the development of this adaptive immune response. So, can anyone tell me what is the difference between innate immunity and adaptive immunity? Anyone can tell what is the difference between innate immunity and adaptive immunity? Hanshika, innate is present since birth, and adaptive we acquire by encounter with an antigen. Yes. So, innate is present since birth, and adaptive we acquire. That is correct. And any other difference? Innate is non-specific and acquired is specific to an antigen. Yes. Anyone else? Anushka or Simran, Lina, anyone can tell. In innate, there are not any memory cells, but in adaptive, yes, we are having memory cells. So innate will be fast or adaptive will be fast. Innate will be fast, but adaptive will take time. Yes. It shows rapid response. Yes, innate immune response will be very rapid, while adaptive immune response will take time to develop. But it will have a memory, but innate immunity won't have any kind of memory. So your adaptive immune system will remember that you have encountered this pathogen before and will act rapidly in the second time compared to the first time. But the innate immune response will be similar in case of the same pathogen first time also and second time also. Okay, uh, so we come to the lymphoid lineage, which regulates this adaptive immune response. So lymphoid lineage cells or the lymphocytes are the principal cell players in the adaptive immune response and is the source of our immune memory. So it represents 20 to 40% of circulating white blood cells and 99% of cells in the lymph. So approximately trillion lymphocytes circulate continuously through our blood and lymph and migrate into the tissue spaces and lymphoid organs. So you can understand so many, there are like so many lymphocytes which are circulating continuously. And if there is a pathogen attack in our cell, they can easily identify and can activate this immune response. So first we come to the B lymphocytes. As we all can see that these lymphoblasts will get divided into B lymphocyte, T lymphocyte and natural killer cells. So first we are coming into the B lymphocytes. So B lymphocytes they develop as well as mature in bone marrow. So bone marrow is a major site of development as well as maturation of B lymphocytes in humans. So these B cells will express a B cell receptor, that is a membrane bound immunoglobulin molecule that binds to antigen. So what happens uh, when there is a attack by a virus or a pathogen? This B cell, which are naive. Naive means they have not encountered any pathogen before. So they are naive, they are new to the pathogen. So when they encounter these antigens, these B cells will become activated. So you can see there are certain receptors which are present on this B cell. We call it as B cell receptor, which are nothing but the membrane bound immunoglobulin. Like antibody, when we call it as an antibody, it is soluble. It can travel to any places. But when we call it as a BCR or the B cell receptor, these are nothing but the membrane bound form of the antibody itself. So this BCR or the B cell receptors will uh, interact with this antigen here. You can see the antigen and it will activate the B cell. So this B cell will get divided into uh, two types. One is the memory B cell and one is the plasma B cell. Then this plasma B cell will produce these soluble antibodies. This membrane bound antibody will then get solubilized and form the antibody which will react to this antigen. So uh, this B cell gets activated whenever there is they encounter any kind of pathogen, and these B lymphocytes, T lymphocytes, all are always circulating through our blood and the lymph, always, so that whenever these B cell will encounter an antigen or a pathogen, they will become activated and they will form two types of cells, that is a memory B cell which will remember that this is the pathogen that I have encountered before, and another type is a plasma B cell which will secrete the antibodies, and these antibodies will go and bind to these different types of antigen. So these B cells can recognize soluble or as well as particulate antigens. So it can directly recognize the antigen or the whole antigens. But in case of T lymphocytes, it can only recognize the processed antigens. So coming to T lymphocytes, they uh, hello. So these T lymphocytes uh, also are formed in the bone marrow, but their major site of maturation is in the thymus. So that is why B lymphocyte, why they are called, because their site of maturation is in the bone marrow. But T lymphocyte, why it is called T lymphocyte, because the major site of maturation is in the thymus. So like the B cell, as I told, this B cell are expressing a type of antigen binding receptor, BCR. There are TCR, a T cell receptor present on the surface of these T cells. So, as I already told, that the B lymphocytes can recognize any kind of soluble or particulate antigen, like small particles, they can easily recognize. But in case of T cell, they can only recognize the processed piece of antigen, typically in the form of very small peptides bound to cell membrane proteins known as I already told you before, like these major histocompatibility complex molecules which are present, they can only recognize these processed pieces of antigen through these MHC molecules. So there are two types of T cells. One is the CD4 plus T cell and another is the CD8 plus T cell. So CD4 plus T cell are the helper cells and the CD8 plus T cells are the cytotoxic cells. CD4 plus and CD8 plus is nothing but some molecules which are present on the surface of these T cells. These helper T cells are recognized by the presence of the CD4 plus molecules which are present on its surface. This, as I already told, there are co-stimulatory molecules which are present on the surface of these T cells, and these co-stimulatory molecules are important for uh, the signaling or the downstream signaling processes. So CD4 plus will be present on the T helper cell and CD8 plus will be present on the cytotoxic or TC cells. So both are different in the case that their one is their marker is different, co-stimulatory molecules are different, and they while CD4 plus T cell will only recognize antigen from the MHC class 2 molecule, CD8 plus T cell will recognize the molecule, the processed antigen via MHC class 1 molecules. So these are the two differences by which these helper cells and the cytotoxic T cells will recognize the antigen. So any kind of antigen presenting cells like the macrophages, the dendritic cell, the B cells will process the antigen and present these antigens via their surface MHC molecules to the T cells, and only then these T cells will become activated and there will be a downstream signaling which will kill or destroy the pathogen. So as we already now studied that the types of lymphocytes are B cell, helper T cell, cytotoxic T cell. Apart from these T cells, there are other types of T cells like the regulatory T cells, and another is a natural killer cell. So there are difference in the antigen recognition by all these cells. Here we can see that the B cell will recognize antigen on the pathogens or the soluble antigen. As I already told, directly it can recognize the antigen, while the helper T cell will only recognize the antigen or the processed antigen which are presented by the professional APCs or antigen presenting cell. In case of cytotoxic T cell, the antigen presented by infected or malignant cell means the cancer cells. It will only recognize the process antigen by the infected or malignant cells. In case of regulatory T cell, no antigen recognition function is there. And in case of natural killer cell, there is self antigen or foreign antigen on host cells. It can either recognize the self antigen or it can recognize any foreign antigen present on the host cell via, and all this you can see are having different types of receptors on its surface. So the effector functions are B cell can, I already told that it can get divided into memory B cell as well as plasma B cell, and it will produce antibodies. So these antibodies can neutralize the pathogens, can phagocytose the pathogen, and it can activate a complement that we will come to later classes. That these are the some proteins which also help in the clearance of the pathogen. And there are this helper T cell which can secrete cytokines and can activate the macrophages. It can activate other T and B cells, and also it can induce inflammation. In the case of cytotoxic T cell, they can eliminate the infected or malignant cells or the cancer cells. And in case of regulatory T cell, they regulate our immune response by suppressing our immune response. Like there should be a balance. Like there should not be any uh, like overactivation of our immune response. That can also lead to damage of many tissues, and it can also lead to inflammation, excessive inflammation. So to balance this, there are certain cells which are known as a regulatory T cell, which can regulate our immune system and can suppress our immune response. Also, it can also help in like deciding that whether the antigen which are being uh, present in our body are self antigen or foreign antigen. Like it should not activate our immune system when there is an encounter with the self antigen. And in case of natural killer cell, they also eliminate the infected or malignant cells uh, by activation of different types of molecules. So, so we have studied about all of these cells, immune cells, but where do these immune cells develop? So, any question about the different types of immune cells? Okay, so now the question is where these immune cells develop. So uh, there is specifically these immune cells are developing in the bone marrow. But when we in a fetus or when a person like during the developmental stage, the site of hematopoiesis changes. Like for example, here we can see that from in human from the 28 to 40 days there is the site of hematopoiesis in the placenta. Then before that it will be in the yolk sac. Then after it will change to the liver. Then finally it will go to the bone marrow. Like there is a shift of this site of hematopoiesis during embryonic development. In both cases of mouse and human, we can see that there is a like it will change depending on the like in the initial stage it will be at a different place, while in the later stage it will be in a different place. So finally before birth it will the site of hematopoiesis will be in the bone marrow. So bone marrow um, is the site where this hematopoiesis takes place. So the bone marrow, here we can see there are different in different types of bones. We are having this bone marrow inside. Here we can see this is the bone and this is the marrow. And this marrow is also known as the medullary cavity, where it is packed with different kinds of cells. So this medullary cavity can be divided into endosteal niche, which is lining the bone, and the perivascular niche, which is lining the blood vessel that run through the center of the bone. So these niches contain some stromal cells that provide structure and guidance for the hematopoiesis. So the stromal cells in this bone marrow will regulate the proliferation, the quiescence, the trafficking, and differentiation of the of the hematopoietic stem cell. So there are different cells present in this bone marrow, like the endothelial cells. Here we can see that lines these blood vessels. Here is the bone. Here is the marrow, which are present, and there you can see presence of the cytokines, growth factors, and here is the blood vessel. And these blood vessels are lined by the endothelial cells, and there are perivascular cells which will interact with these endothelial cells. There are certain nerves which are sympathetic nerves that will transmit the signals to the other cells. There are macrophages which will uh, influence the activity of these other cells, and there are osteoblasts which will generate the bone cells. So there are, here you can see different types of cells which are present, which are having different kinds of function, and through the blood vessel, this there this B cells and T cells, B cells and T cells can move through different paths. So another is the thymus. So primary lymphoid organs consist of bone marrow and thymus. So as I already told, this thymus is the site where these T cells or the thymocytes mature into functional T cells. Here we can see it is the bone marrow is present and thymus is present just above the heart. Here is the diaphragm, here is the heart. So here we can see that this thymus is present, and thymus also contains cells, mature functional T cells, which are known as thymocytes. These thymocytes, whose, like, there are negative selection as well as positive selection inside the thymus, which are happening. As we can see, the thymus is also divided into two parts, like the medulla and the cortex. And this medulla and cortex are full of cells, and there are processes which are happening inside the thymus, which are known as negative selection process and positive selection process. Like, negative selection is when these thymocytes, which whose these T cell receptors, which bind to self-MHC peptide complexes with too high affinity, are made to die. So there will be generation of too many cells in the thymus, but most of the 95% of the cell will undergo death. There, which will undergo death. The thymocytes, whose these TCRs bind to the self MHC or peptide complexes, like our self antigens, if it is reacting with the TCRs, then these cells will die, because this can lead to complication. Like, if our own cells are binding to our own antigen, then there will be development of disease known as autoimmune disease. So these cells which are with too high affinity are binding to our self MHC or peptide complexes, they will die through a process known as negative selection. And the thymocytes, which will bind to these peptides, which are very low affinity also, we are induced to die. Only the thymocytes that will bind to self-MHC peptides with intermediate affinity undergo positive selection and mature into the thymic medulla before entering the circulation. So there should not be too high affinity or too low affinity. There should be a intermediate affinity with which this TCR should bind to the peptide MHC complex. Only then it will undergo maturation and will migrate to the thymic medulla before entering into the circulation. So almost 95% of these thymocytes die and 5% only survive after this surveillance. So this T cell precursors enters the thymus in blood vessels at the corticomedullary junction between the thymic cortex and the thymic medulla. So here we can see, like, here it is the thymus, and there are different parts like the cortex, which is the outer region, medulla, which is the inner region. So this T cell precursor will enter into the thymus in the blood vessels, at this junction, which is known as the corticomedullary junction. Here, double DN means double negative cells, DP means double positive cell, and SP means single positive cell. What does this mean? As uh, I already told that there are two types of T cells. One is T helper cells and one is T cytotoxic T cells. So T helper cells are having CD4 plus molecules on their surface, while T helper cells, T cytotoxic T cells are having CD8 plus on their surface. So the CD4 plus cells as well as CD8 plus cells, first when a T cell is entering into this thymus, they are having neither, not CD4 plus nor CD8 plus. So they are double negative. So they are not having any CD4 plus or CD8 plus cells on their membrane. So they are these cells with no CD molecule CD4 or CD8 plus are known as double negative cells. So they are entering into the thymus. First, they are going into the subcapsular cortex, and the subcapsular after the subcapsular cortex, they will proliferate first. They will undergo proliferation and will enter this cortex where they will express this mature TCRs. But here also what will happen, they will become double positive. That is once TCR, one T cell will be having both the CD4 plus as well as CD8 plus. So it's double positive cells. So after these double positive cells are forming here in the cortex, they will then go to this uh, thymic epithelial cells, and after then when they enter the medulla, they will be segregated into single positive cells, like where they will have either the CD4 plus or either the CD8 plus. Those TCRs which are having both the cells, they will die. They will undergo a death process, but the ones which are having only the CD4 plus and CD8 plus will be selected, and here only they will segregate either the cytotoxic T cell or T helper cell. So in this medulla, they will they here only this maturation takes place, and they will go to the medullary thymic epithelial cells here, and they will go into the circulation. So this is a very important point. Like this, T cells have to get matured, and it has to be either CD4 plus or CD8 plus positive. If it is like it is having both CD4 plus and CD8 plus, it will undergo a death process. So the selection of these T cells happens in this cortex and the medulla of this thymus, and this maturation also occurs in this thymus, from where it goes to the circulation. So after this, there are these are the primary lymphoid organs at the thymus and the bone marrow, and after this comes the secondary lymphoid organs. So primary lymphoid organs is where the these B cells, T cells are generated, and secondary lymphoids are what they where the immune response is initiated. First, these B cells, T cells will get developed, get matured in this bone marrow cells as well as the thymus. Only then will comes the secondary lymphoid organ where they will encounter these antigens. So, any question till now? Anyone is having any question or any doubt? Someone has newly joined, Gunavati.
Yes, ma'am.
Yeah, so hi, from where you are, you can introduce yourself.
Hi, I'm Gunavati from St. Peter's Research Institute and College.
From?
I'm working as assistant professor in microbiology.
Okay. Okay,
Results, no problem, ma'am.
okay, thank you. Okay,
Thank you.
um, so after that, this we come to the secondary lymphoid organs where this immune response is initiated. So, so this secondary lymphoid organ consists of a lymphatic system, which is a network of vessel filled with a protein-rich fluid, which we call as lymph, which is derived from the fluid component of blood plasma. So here we can see that our lymphatic system is distributed all across our body in different parts of our body. Like here in the tonsil, in the this is the right lymphatic duct. This is the thoracic duct in the spleen, and here also we can see that lymph nodes are present. The tissue lymphatic system is also present here. So it is distributed throughout our body, and whenever there any antigen, there is a antigen which is entering into our body. This that antigen will traverse to all over this lymphatic system where they are recognized by our uh, lymphocytes, which will then start that immune response. So here, so lymphatic system basically is there are two types of fluid. Like blood we already know is a kind of fluid. So apart from blood there is another fluid, that is the lymph, which is flowing through all through our body. And this lymphatic system and blood are the two important uh, system that actually helps in initiating this immune response in our body whenever there is any pathogen attack. So the walls of these lymphatic vessels are thinner than those of these blood vessels, and are more porous. So anytime there can be a uh, the cells can easily enter into these lymphatic vessels, and they can encounter these pathogens inside this lymphatic system. So they are consisting of a single layer of loosely endothelial cells and allow fluids to and cells to enter into the lymphatic network. So both the T cells, B cells, or the antigens can easily enter into our lymphatic system where this when there is an interaction between this lymphocytes as well as the antigen. So the lymph node which is present in our lymphatic system is a highly specialized secondary lymphoid organ. So this lymph node is also having two regions, that is the cortex as well as the medulla. The cortex is the outer region, medulla is the inner region, and it contains lymphocytes. The cortex contains mainly lymphocytes, mostly B cells, macrophages, and follicular dendritic cells arranged in follicles. And beneath the cortex, the paracortex. Here we can see that the cortex is present, which is consisting of this mostly this lymphocytes, the macrophages, and there is paracortex, which is present. This paracortex is also called the T cell zone, and the B cell zone or the follicle is known as the B cell zone, where these dendritic cells are found. And in this paracortex or the T cell zone, there are presence of a large number of T lymphocytes that also contains the dendritic cells, and these dendritic cells and T cells have migrated into the lymph node from the surrounding tissue. Here we can see that there are efferent uh, as well as afferent lymphatic system from where there is entry of these cells into this lymph node, and these cells can exit this lymph node, be it cells or the antigen. They will enter into this lymph node through the afferent lymphatic system and will exit this through the efferent lymphatic system. So this medulla is the innermost layer where the lymphocytes exit the lymph node through the afferent lymphatics. So from here we can see the nave lymphocytes are entering, and here from the afferent the antigens are entering, and upon encounter of these antigens by this nave lymphocytes, they undergo a clonal proliferation. Like they will encounter the antigen and they will clonally get selected. The antigen will react to the receptor um, present on these lymphocytes, and they will clonally get selected. Means suppose there are 10 types of lymphocytes which are present. Up with this 10 in out of these 10 lymphocytes, if the antigen is reacting with one uh, lymphocyte which is having a specific receptor, then that particular like lymphocyte will get selected and will proliferate to make a large number of lymphocytes. So then they can after the proliferation they will then reach out and then out like leave this lymph node through the efferent lymphatic system. So here we can see these are the microscopic images like the B cell follicles and the T cell zone and the paracortex. And this is having the germinal center where the large number of cells are present, the mature cells are present. Uh, so, so apart from the lymph node where this interaction takes place, another major secondary lymphoid organ is the spleen. So the spleen is present here, we can see just below the heart, here it is present. So the spleen is organizes the first immune response against blood-borne pathogens. So the spleen also is compartmentalized into the different zones, which is the white pulp and the red pulp. The white pulp here we can see that the white pulp contains the majorly the B and the T lymphocyte, and the red pulp which contains the circulating red blood cells. And here also it is having its own artery, its own vein, and similarly it is having different types of zones like the marginal zone, the follicle zone, which is having the B cell, this white pulp zone, which is having the T cells, and these sinuses are also present in between, and this red pulp zone which contains this red blood cells. So the periarterial lymphoid sheath of the white pulp includes B cell follicles and the uh, T cell zones. The splenic marginal zone, here we can see that the splenic marginal zone, which is a specialized region, contains the macrophages and B cells and forms a boundary between the red and the white pulp and plays an important role in trapping and response to blood-borne pathogens. So this is the first the first immune response against pathogens which are blood-borne, which are traveling to the blood. So this spleen is the first site where these blood-borne pathogens are trapped. So it is also an important secondary lymphoid organ. So in people or the children who have undergone splenectomy, that is in uh, children where the spleen has been removed by surgical means, they are shown to be vulnerable to different types of infection like streptococcus pneumoniae, hemophilus influenza, different bacterial infection they are very much susceptible to. So so we can understand that how spleen plays an important role against this blood-borne pathogens, especially these bacteria. So apart from that, there are other organs also, like which are present, which are barrier organs, like which include our skin, which include our mucosal tissue, which also contains the secondary lymphoid organs tissue, and they also like mount an important defense against pathogens that penetrate our epithelial layers. For example, there are tissues which we called as gut associated lymphoid tissue, or as mucosa associated lymphoid tissue. This lymphoid tissues also plays an important role in mounting an immune response. Like whenever there is a infection which is going through the skin, or suppose which is going through the our mucosal surface, or through our gut, like they 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 also can easily recognize this infection and can mount an immune response. They so uh, Here they also contains this epithelial cells, and which play an active role and initiate the response of innate and adaptive immune cells, and which can organize into B cell follicles. And they also help us to tolerate uh, gain tolerance against commensal microbes that coexist at our surface. For example, our gut is exposed to a large number of microbes. Like different types of microbes are present in our gut, which help in our digestion and other processes. But our immune system knows that uh, those microbes are actually not pathogens, or they will not create any harm to us. So this immune system, or the gut associated lymphoid tissue, uh, will help us to maintain some tolerance that these are commensal, or the microbes which are helping us, and not any kind of disease causing pathogens. So they will maintain a tolerance against the commensal microbes also. Or sometimes whatever food we are eating, suppose we are eating some food, but those food are kind of antigenic to us, like those food are kind of foreign substance to us, but our mucosal surface knows that like these foods are not foreign to us. Like they are not any kind of pathogen, so they maintain a type of tolerance. Like these, they know that these are not going to harm us, so they kind of maintain a tolerance to these all these kinds of microbes, or any types of food which you are intaking from outside, and help us to maintain like a balanced immune response. So here we can see like there are different types of cells, that is the epithelial cells, and these are the bacteria which are there. There are inside the epithelial cells, there are specialized cells called the M cells that convey antigens from the intestinal lumen to the inner layers of the intestinal wall. So they help, they trigger the formation of B cell follicles. Here we can see that they will trigger, like when the bacteria is entering, these M cells are allowing this bacteria to enter into this enter inside, and then they will trigger the formation of these B cells, and these B cells will then divide into the plasma cells as well as the memory cells, and these plasma cells will secrete the antibodies. These antibodies will then again enter into the lumen. Here we can see that it is entering into the lumen, the antibodies, and then will bind to the pathogens, protecting the intestinal wall from inflammation and invasion. So other cells also are present, like macrophage dendritic cells. They will also uh, they will also encounter these pathogens from the lumen, and with the help of these regulatory T cells, they will work in understanding or distinguish between the what are the beneficial commensal bacteria and more dangerous pathogens. So antigen uh, presenting cells and lymphocytes will then travel to the local lymph nodes, where they will trigger a more systemic immune response to antigens. So here we can see that how the commensal bacteria and the pathogens can trigger different types of immune response, and how these uh, B cells are getting like known that these are the commensal bacteria and these are the pathogens, and they are mounting an immune response. So when they are encountering an antigen, this if, the B cells, T cells, or other antigen presenting cells will migrate via the lymph nodes, and they will trigger a more systemic immune response. But whatever bacteria are already present in our gut, they know that these antigens are known to us, so they won't this mount this response and act against the pathogen. So this is how the tolerance should be in our immune system. They should know what are the self antigens, what are the commensal bacteria inside our system, and accordingly they should react to the pathogens which are entering into their body. So if there is an excessive immune response and there is a reaction of this immune response against our own pathogen, own bacteria or own antigen, then there will be excessive inflammation, and that can lead to many diseases like autoimmune diseases, or in case of other extreme conditions there can be too much production of cytokines or cytokine storm, which can lead to even death. So the our immune system must know to distinguish between the beneficial as well as the dangerous pathogens. So that's all. um, so anyone is having question any question, whatever. Everyone understood everything? So no one is having also doubt, like in any sir's presentation for week 1 and week 2. Have you all gone through the assignment? So Simran, can you tell me what are the primary lymphoid organs and the secondary lymphoid organs in our body? Or Lina, anyone can tell primary or secondary?
Ma'am, primary lymphoid organ includes bone marrow and thymus, whereas the secondary lymphoid organ will include lymph node, spleen.
Simran? So, what is the site of maturation of B cells and T cells?
Bone marrow and thymus.
Okay, and formation of B cells and T cells? No, formation. I am telling like where these B cells and T cells develop. Site of maturation, I understood that B cells and T cells are maturing in bone marrow and thymus, but where is are these B cells and T cells are forming? No, hematopoietic stem cell is the cells from where these cells are forming, but I am asking which organs they are developing, in which organs. Simran, where is this bone marrow? Where is this B cells and T cells are developing? Which organ? No, bone marrow and thymus is the place where they are getting maturation, but where is the development? Like from where they are originating? Actually, you both are wrong because in bone marrow only these two type of cells will be formed, but maturation will take place differently. T cell will also be developed in the bone marrow only, but it will leave the bone marrow and will enter into the thymus where its maturation takes place. So because of the site of maturation, they are getting the name like B lymphocyte, T lymphocyte. So bone marrow is the site where both these lymphocytes will be formed, but site of maturation is different. T cell will mature in the bone marrow while T cell will mature in the thymus. So this hematopoietic stem cell will be only in the bone marrow. It won't be in the thymus. So both of the cells will develop in the bone marrow only, but the B cell will mature in the bone marrow, but T cell will leave and will mature in the thymus. So the name is T cell. Depending on the site of maturation, the name has been given. Uh, did you guys understand what I am telling? Okay, so any further question is there? Okay, so you can, if any doubt is coming regarding solving of the assignment or any kind of in any lecture like week 1, week 2 or week 3 coming week, then you can also mail me or you can like write in the portal if you are having any kind of doubt. So thank you. Thank you for listening.